Okay, so good evening. Welcome to today's session. This is our 10th session for this course, and we have two more sessions left actually. If required, we might have an extra class, but we'll decide that based on how the extra classes go. But for today, welcome to today's session. We will start off with a quick recap of the last week because we're running a little late today, and then we'll move on to the discussion for today. We'll end the class as pre-planned at 7, but whatever is left over in case there's anything, I don't think it should be a problem. We'll wrap it up in the next class by extending that. So, just to remind everybody of what we were discussing in the last class, we diverted a little bit from the originally discussed flyer, and we had moved towards the use of different types of models, which can be used to sort of represent the condition in patients within the lab. And we discussed in vitro and in vivo models, which are all used extensively in the field of cancer research by scientists. And we started off with a quick recap of these in vitro and in vivo models. So we talked about cell lines, organoids, etc. And then we discussed about in vivo models as well. These were some key differences between the two. I'll not go over them because that will be a little redundant. So you can look at that in your own time. We went through a comparison of all of these different systems, taking into factor how easy it is to establish, the cost that is actually associated with the establishment process, the type of experiments that you can actually host in these systems, and how closely they are actually able to recapitulate the condition within an individual, within a patient. Then we also had looked at how useful that particular system can be at genetically manipulating it. And when I say genetic manipulation, it means that if you want to characterize one particular mutation, how easily and how accurately are you able to do that? That if you're suppose you want to mutate gene X, you have to be able to confirm that only gene X is mutated and the overall genome of the organism is not somehow affected by that mutation. So the specificity and the accuracy of your experiment can differ based on what type of model you've actually used. Then the physiological complexity, the relative cost, and of course the recapitulation of human physiology. So all of these factors have to be taken into mind. Then we had quite an extensive discussion about how you can establish 2D models for cancer research, which of course most predominantly are cell lines, especially because they are providing an indefinite source of biological material to your lab for experimental purposes. And the way in which they're established is that you isolate cancer cells from patients, and then they are grown in a specifically defined culture media, which can be then also transplanted into immunocompetent mice, and then you generate xenografts for the same. Xenograph basically means simulation of a tumor within an in vivo model for your experiment. And this stabilization is possible because these cells are immortalized and they are like they have overcome the barriers of senescence and they are able to replicate indefinitely and they can be passaged if you maintain the growth conditions. However, I'd also mentioned that from time to time, you actually need to get their profiling done so that you can ensure that whatever genomic profile is expected from that cell line or is commercially and globally validated, that is also retained in the cell line that you are using for your culture. So as to ensure the accuracy of your data and whatever you report can actually be true for any person across the world who is trying to replicate the same. Then we discussed a little bit about the HeLa cell line, and I'd asked you to go and read up a little bit on the bioethical dispute about the establishment of this cell line, and also how it has been used as a model. And in fact, if I've not mentioned it in the previous class, HeLa is one of the cell lines that actually needs to be handled with a lot of care. It is extremely easy to culture. When I say easy, I mean it's very uh, sturdy as a cell line. It does not need to be handled with a lot of care because it has such intense proliferation capacity that you can handle it very easily, and it will be able to even from a very small number of cells, you'll be able to have a confluent dish really soon. Confluency, if I've not mentioned it before, basically confluency is the if you if you're looking at any particular culture dish, say for example, this is the view you are getting from your microscope, then of the total area that is visible, your field of view, from that entire field, the area which is occupied by cells indicates its confluency. So this is, while it is written as low density, it's actually low confluency. That's the more correct scientific jargon for this purpose. And this is high density, which means that more number of cells are populating your field of view. And it is a term which is used very commonly in cell culture. Anybody who has handled cells would obviously be pretty familiar with it. And basically, the reason for at all being bothered about this term is because how confluent your cells are can actually dictate how you can perform your experiments. So, for example, if you want to do some sort of a drug treatment, then it is always better to go for a confluency like this, where the cells have some space between each other, so that whatever drug you're actually treating your cells with, that gets access to cells independently, and like there's enough space for the drug to be able to enter into the cell. However, if you have clumps of cells like this, which are all present in really dense gatherings, then you cannot always ensure that, okay, a drug has entered cell X and cell Y and cell Z. And as a result, when you eventually want to study the effect of that drug, which is usually done using common molecular biology techniques like Western blotting or qPCR, for example, quantitative PCR, for example, then in that case, you will actually have data from a mixed population, half of which might have undergone treatment and half of it might not actually have undergone the treatment. So whatever effect you are supposed to get from that drug treatment might not actually come out to be true, even though experimentally you have made no errors. So whatever you report might actually the accuracy or the correctness of your report might actually get compromised if you do not ensure all of these little things. So these are all things you learn as part of your job, and it's all covered when somebody is handling cells for a definite period of time. Another cell line which we understood here as an example was the Raji cell line, which is for Burkitt's lymphoma, and that was the first human continuous hematopoietic cell line. And we had also looked into this factor that copy number alterations and transcriptional profile of cancer cell lines, they are expected to show numeric alterations pretty similar to that of primary human tumors, which is actually the advantage part of using cell lines. However, it's not all idealistic, and we can find a lot of disadvantages associated with the same as well. First of all, being the lack of heterogeneity. Within the cell population, heterogeneity is quite compromised. Not compromised, it's minimal. Let's put it that way. It's not expected to have heterogeneity. Therefore, as a model, it lacks certain features which might actually be useful for your study. Then limited representation, because not every subtype of every cancer can actually be modeled using cell lines, simply because they do not exist. Of course, there's scope that in the future you'll have more and more cell lines. But as you can understand, for a cell line to actually be established, to be commercially viable, and to be standardized to such an extent that it is truly considered a model for research, that's going to take a lot of work. And people have now moved from that idea and have diverted a little bit more towards the 3D models, which is what we discussed during the later point of the class. So therefore, this cell line panel obviously is still quite lacking. Then the lack of stromal components, which are the associated cells present in the extracellular matrix, all of these are also lacking. And finally, the nature of these cells' growth is also something which is limiting and does not truly replicate the physiological dimensions of a tumor. Just excuse me for a second, it's beeping. Okay, so these were the disadvantages of cell lines, which eventually caused the shift towards the patient-derived 3D models for research. And these were two examples of organoids that we saw. This one was for cervical organoids, this was for a cerebral organoid. And then we looked at the historical timeline for the same. I'll not go over it again in too much detail, but basically we started out from cell lines, moved towards spheroids, and patient-derived xenografts. Then eventually people moved towards microfluidic devices as recent as 10 to 15 years ago. And now tumor on a chip model is something which has been extensively studied and is aiming to be commercialized. Yes, all of this needs a lot of time before it can be truly used as a model for all sort of oncology research. And in fact, before it can be made affordable for all sorts of labs. So all of that is going to take a lot of time. Then we drew a comparison between all of these 3D patient-derived models, starting off with, again, 2D cell lines, the organ on a chip concept, which I briefly explained in the last class, the spheroids and organoids, and finally rodent xenografts as well. As you can see, each of them had a couple of advantages associated with them, and of course, they also lacked certain features leading to their disadvantages. Like we discussed for 2D cell lines, then the organ on chip, spheroid, organoids, and xenografts. So despite these pros and cons, you obviously need some sort of model for study. So depending on the aim of the experiment and the type of hypothesis you are chasing, the model that you use for your study can vary. And overall, each of these models are acceptable. It just depends on the purpose you are trying to satisfy, the type of model you will choose. Um, then we had discussed a quick chronological recap of organoids, the type of organoids which have been established, and where the roots of the same were sown, majorly focusing on the discovery made by Professor John Gurdon and Professor Shinya Yamanaka, where they discussed, or where they actually brought to the world for the first time the concept of human-induced pluripotent stem cells from adult stem cells, and they showed how you can actually generate all types of almost all types of body cells of a particular individual from their stem cell, and that stem cell is actually resulting from the manipulation of an adult somatic cell, and therefore the concept of iPSCs was brought forth, and that has actually revolutionized the field of organoids. And as you can see, back to back, every few years a new type of organoid has been established and is used for study, and of course it's been an immense contribution to the field of biology. Um, we had ended on this slide where we made a comparison between different types of in vivo models for cancer research. We discussed about different types of immunocompromised and immunocompetent mice. Majorly here, the comparison is made for immunodeficient or the not skid and the skid mice, or the nude mice, or the not skid gamma mice, and the RAG or NRG mice. All of these different types of models were established. And I think I showed you the portal as well, how to access it, how to, not the portal, but the type of companies which have actually laid the groundwork for all of these immunodeficient models of research, majorly two companies, which are Charles Rivers Laboratories and Jackson Laboratories, and the different type of information that you can access from each of these websites, like the genetic background, the type of immune components that they actually lack, and what sort of experiments can actually be performed for them, the difference in their average lifespans, and all of those subtle differences. We discussed all of that in the last class. And with that, we had pretty much ended that session. With that, we'll move towards today's session. If there were any queries from the last class, you can unmute at any time and let me know. But yeah, I'm rushing a little fast as obviously we're running a little late today, so I'll brush through topics quickly. Okay, so today's theme is actually going to address this last part of the flyer. And in the next class, I'm aiming at completing these key parameters used in oncology research. But for today, our theme is to learn a little bit about cancer evolution, learn a little bit about drug resistance, and how to actually deal with it. So, first of all, this slide is showing you an example of cancer evolution. So just start off by looking at this sort of time scale, like how do I explain? It's a schematic. It's not actually representing anything, but basically it is showing you how from the central population of cells, how cells can actually diverge into different directions. And as you can see, these arrows, they indicate that there is some interchangeability or some sort of transformation which occurs in a reversible manner, and that can go in either direction. So let us go through it one by one. But before that, let's understand the definition. So, what is cancer evolution? So, the process by which cancer cells adapt and they survive by the acquisition and selection of molecular modifications is defined as cancer evolution. What does that mean? It means that based on what sort of condition you have and what sort of modifications are helping you adapt the best to that sort of condition is part of an entire process, and that process is what is known as cancer evolution. So let's again come back to the schematic. You can see three different components or three. Oh, just a second. Yeah. So as you can see, there are three different components. First of all, you are seeing that there is an emergence and selection of genetic variants. Then you see there's cancer cell plasticity, and third, the tumor promoting microenvironment. Yeah, the tumor promoting microenvironment. So what is it? What is that exactly implying? It means that from your central population, you can see that cancer cells can evolve in different directions by acquiring different types of characteristics. If you have gone through the initial slides carefully, you would see that these terms might seem a little bit familiar to you. When we had first discussed hallmarks, we had talked about some of these, and I had asked also everybody to go through the differences between core hallmarks and enabling characteristics. That's actually going to be very relevant here. But even if you haven't, you basically can just conceptually understand it, that each of these are sort of characteristics or traits which cancer cells can acquire over time, and that is going to define how they are going to eventually progress. Like, for example, if cancer cells acquire the ability to have a lot of genomic or genetic instability, what they're eventually going to result in are genetic variants. And of those genetic variants, those which respond or those which survive the best in presence of the drug or in presence of whatever XYZ condition is surrounding them, those will be selected, and therefore they will they will be preferentially selected against all of the other variants, and they will take over the other cell lines, based other cells or other genetic subtypes. On the other hand, if they acquire the ability to undergo a lot of epigenetic reprogramming, then you can see gradually what is the first most obvious change you can see. You see that the cells start becoming different in their shape, and also you can see that there's emergence, the color keeps changing gradually. And over time, each of these things is very important. Like, do not take it lightly. Keep a very keen eye on this. If you look at the first branch, you see that the cell shape is overall maintained. But what you see are very clear differences in color. That is because they are all genetic variants of each other. As a result, whatever genetic mutation has occurred, that has led to an overall change within the same. However, when you look at epigenetic reprogramming, what do you see? You see that the cell starts transforming, it starts changing. There are intermediate steps as well. And finally, the population which gets selected or which predominates is extremely different in shape from the first one. And it has also changed this color, which means that it is carrying some other the differences in the overall transcriptome as well because of the epigenetic reprogramming that has occurred. And this ability of cells to undergo this change, where there's a gradual shift from one type to another type, and usually that is the result of some sort of epigenetic reprogramming along with other changes as well. That entire process leads to what is called as cancer cell plasticity. Plasticity means that they can transform or they can alter from one lineage or from one subtype into other subtype, and you can obviously find some intermediate stages as well. So this is another way in which cancer evolves. So you will find all of these distinct sub-steps or transitional steps in this case. The third one is resulting in a condition where the cancer cells are sort of undergoing changes or they are evolving over time as a result of interactions with the tumor microenvironment, as a result of which they are eventually undergoing evolution in such a way that they are changing the environment surrounding them, which helps them grow better. So instead of changing themselves, cancer cells are secreting certain factors, or they're able to control the surrounding microenvironment in such a way that now it favors their growth instead of opposing them. So that is the result of tumor and microenvironment interaction, which again was a part of the hallmarks discussion that we had in the very initial sessions. And yeah, these are basically the three sort of ways in which they can usually undergo transition. But also, what you need to keep in mind is that there is some sort of interchangeability between the two as well. That sometimes within the environment, microenvironment, which is tumor promoting, there can be some sort of acquisition of some, genetic differences or genetic variants, and they can get selected against and selected for, and they can then populate. These cells can also undergo this transition and lead to plasticity. These changed or altered transdifferentiated cells. Transdifferentiation is part of that process where the cells gradually evolve from one lineage into another, like I just explained. And they can also interact with the tumor microenvironment, and then they can also manipulate it according to their requirement. So this is basically the legend for this figure. This paper, you can refer to this paper. It's a beautiful paper. It's a review article where they have discussed a little bit about cancer evolution, and it's beautifully explained. It might be a little advanced language-wise, but if it is interesting to you, it'll be a great read. So, what they have discussed are the many phases of cancer evolution, enabling characteristics, like I mentioned, such as genetic instability, epigenetic reprogramming, and interactions between tumor cells and the TME, enable cancer evolutionary mechanisms, such as the emergence and selection of genetic variants, cancer cell plasticity, and the re education of the tumor microenvironment. So, these are the three major subtypes. Please let me know if this was clear, if there are any queries, anything you want to discuss, please let me know and then we move on.
No, ma'am, it was very clear.
Okay. Okay, if Harsha, if you have any questions, let me know. Okay, so now moving on, the question that you might now address is that how is cancer actually evolving? How is it going in these different directions? We talked about the different sub branches. So historically, what has been studied and what is pretty much acceptable across the world is that tumorigenesis, or the process of malignant progression. Again, these are very important terminologies. Please make sure you understand the differences and the implications of each of them. But basically, yes, tumorigenesis means that the onset of cancer formation, or the beginning of cancer, or the changes which are helping cancer form. And then its malignant progression means all of these different substeps, like we just discussed, the substeps as cancer becomes more and more aggressive, and it eventually forms a much more lethal tumor. That entire process embodies a Darwinian process of dynamic evolution. What does it involve? It involves selective pressures encountered by proliferatively expansive outlaw cells. What are outlaw cells? Basically, obviously, as the name suggests, these are cells which do not obey the law, right? So these outlaw cells, they face certain barriers, like barriers and limitations, which are normally physiologically intended to preserve tissue homeostasis, which means that they are responsible under normal circumstances to preserve the condition of the cell and to prevent the emergence of inappropriate phenotypes, right? That is what you want in a normal cell. However, these outlaw cells are able to face those, and they are able to bypass it, and they can then, progress despite all of those things. And that falls under the purview of dynamic evolution, as was very long ago elucidated by Darwin. So, this stepwise evolution that ultimately circumvents these protective mechanisms is apparent in epidemiology and histopathology characteristics of human cancers, which means that how it progresses, how it eventually is able to bypass and is able to circumvent these protective mechanisms, that these checkpoints that are placed within a cell, that has been studied extensively, not just by the histopathology, like again, if you remember, uh, okay, maybe I'll just take a quick tour back. very early on, we had discussed this. You remember how we had seen that from mild to moderate to severe, there can be different staging. All of this is extensively done in histopathology and is done till date. It is something you have to, like if a patient is undergoing, some sort of diagnostic procedure where they're investigated and they're being checked extensively to make sure that the diagnosis is correct, you will definitely have to undergo eventually a patient biopsy, and then the biopsy will have to be sectioned, and then it has to be stained, and then it has to be observed, and then a clinical pathologist is going to finally annotate that yes, there is X type of tumor, Y type of tumor, what should be the prognosis, whether or not that patient has any chance of survival or not, all of those things. So histopathology actually takes into factor how these different substeps evolve, because if you take periodic biopsies from a particular patient, say every 6 months, if someone is undergoing a biopsy, you would be able to actually see how cancer has evolved. And that stepwise evolution, like is mentioned here, it can be studied quite easily. And the same thing can also be observed in the epidemiology of human cancers, which means that if you take population data, you'll be able to see different gradations and different types of tumors within a population. Yeah. So this is quite apparent. These stepwise evolutionary steps are quite apparent in the histopathology of human cancer, and also in the realization that distinctive capabilities dubbed as the hallmarks of cancer actually underlay the process of tumor progression. This we are already familiar with. The appearance of such hallmarks can be seen as the result of an evolutionary process. Sorry. Tumor phenotypes that can be considered as enabling characteristics like genomic instability and epigenetic reprogramming, like we just discussed. They facilitate tumor cell diversification and the Darwinian selection of the set of hallmark traits which are instrumental for these processes. Basically, it means the same things. These two enabling characteristics, which are genomic instability and epigenetic reprogramming, they are extremely relevant to the field of cancer evolution and resistance, because they actually help in increasing tumor cell diversity, help in increasing genomic instability, and therefore, the more diversity that exists, the better is the chance that one of them will be a better survivor or will be selected for in a particular background. So that's what you see here. And here in the schematic, you can see all of the different types of this is a schematic representation of all of the ways in which genomic instability can actually be observed. So what are the different types? The first obviously is a double strand break. As you can see, there's a complete breakage of both of the chains or both of the phosphodiester backbones from both of the helical chains in a particular DNA double helix. Or there can be a single strand break, which is often also called as a nick, which means that it is just a break in one chain that has a much higher chance of repair. Um, why? Because obviously you have the complementary base and the rest of the covalent bonds holding it together. Third, there can be sometimes there can be DNA crosslink whereby some bases, sorry, whereby some bases they actually can form these sort of interactions with each other, where they can form DNA crosslinks. Then you can see there can be base mismatches, insertions, deletions, and base alkylation. If you've studied DNA damage repair before at some point of time, you'd already be familiar with it. But yeah, these are basically the type of genomic insults which can be most commonly seen for any cell. And of course, very prolific, very necessary systems are put in place within a normal cell to make sure that any cell which carries a lot of these alterations does not eventually proliferate, because that is going to lead to a lot of loss of correct genomic information or random changes, or basically it disrupts the integrity of the genome. And those systems are called as DNA damage repair systems or DDR systems, which briefly have been explained here. Like there's a mismatch repair system, which is responsible for correcting base mismatches present in the genome. Second, you have these base accession repair, which is often also called as direct repair, whereby there's a single change or single modification in a base, and then that is removed or that is corrected. Like for example, there can be a base alkylation, so that can be corrected. Then you have the nucleotide accession repair, which is responsible for removing these kind of DNA adducts, which are also called as DNA crosslinks. And sorry, this was direct repair. This is base accession repair. So there can be a single base change, and then that can be altered. Um, Double strand breaks are repaired by different sort of pathways, but mainly by homologous repair pathway, which has a major role to play. So these are all of the different checkpoint and repair mechanisms which have been put into place, and they're necessary for the normal functioning of a cell. Why? If if you study a little bit about this, if it is interesting to you, it's a beautiful field, DNA damage repair. You will find that mutations in the proteins that are actually part of these pathways, they are leading to very, very severe diseases. Let's just take one very direct example. You can see from here that as part of the homologous repair or HR pathway, you have BRCA1, BRCA2, RAT51. I'm sure most of you would be familiar with BRCA1 and BRCA2. They have been extensively like popularly recognized as genes which are used for actually screening patients, that if patients carry mutations in these genes, they have a very high propensity to eventually develop breast cancer. And not just breast cancer, as you can understand, because they are mutations within a pathway necessary for normal cell regulation, which means for normal repair, normal homologous repair within the genome. So if BRCA1, BRCA2 are mutated, and for that matter, if both of them are mutated, it's going to be a horrible situation. And it probably will lead to very early onset cancer. But that's usually not the case. Usually you find mutations in only one gene. But yeah, if somebody carries a mutation in this gene, then they're advised to undergo some sort of precautionary treatment, because they very, very high chance exists that they probably are going to eventually develop some sort of cancer in the long run. So this is one of the reasons why people undergo this sort of screening, especially if somebody has a history, a family history of cancer, and then you can figure out whether there's a genetic component actually attached with the same, or if it is just an adult onset disease that develops because of lifestyle changes. Somebody has faced a lot of exposure to ionizing radiation. Someone has been exposed to a lot of maybe radioactivity, maybe some sort of a mutagen. These are different situations, but yeah, these are like the ones I'm discussing right now. They are genetic defects which eventually can lead to cancer. So yeah, and you can go through these in your own due time, because this is again a very long discussion. I might take a course on this in the next semester, but it's not decided as of now. Let's see. It's a very, very interesting field, very interesting and very relevant. It has been studied extensively, but very relevant in the long run. And you can refer to information about these from the proteins which are associated with them. You learn a little bit about, or you must have heard a little bit about PARP inhibitors, so you can see what pathways they are a part of and how inhibiting them can actually be very good for targeting cancer cells. But of course, that's not desirable for a normal cell, but for a cancer cell, that might be a wonderful way of shutting them off. So yeah, that was the basic idea. But a key attribute of genomic instability is the fact that, first of all, let me just revise what is written here. So beyond tumor initiation from a single founding cell, it's very important to recognize that cancers continue to undergo the process of Darwinian selection, and therefore they continue to evolve. This is something we've already discussed. So here you have a healthy cell within which, just see here, you can see there's a genome also, which is intact and which is healthy. Now it has, yeah, now it has undergone some sort of exposure to some sort of carcinogen. As a result, because of that exposure, the carcinogen is induced, say, mutation X. That is leading to an alteration in some part of the genome, which is rendering it as the first driver mutation that the cell is actually tolerating. Now that initiates some precursor with some stem cell-like properties. So you can also see a timeline, a parallel timeline running with the same, whereby you can see there's this first and initiating mutation, and then that is followed by the acquisition of some cancer hallmarks, like we just explained previously, and that along with additional driver mutation shown here in yellow and certain passenger mutations shown here in green and blue lead to the formation of the first founding cancer cell. Further genetic evolution, which involves more and more mutations, more and more emergence of subclone. So, like you can see, from one subtype, many different or from one type, several different subtypes have emerged. And of them, the predominant ones would be better at adapting to their surrounding condition, and therefore they would be lead to a lot more severe disease. So this is what it is eventually also going to lead to is a very genetically heterogeneous type of cancer, because depending upon what type of driver and what type of passenger mutation that cell has actually tolerated, eventually the mass of cells or the clump of cells can actually be very genetically diverse. And therefore it is A, very difficult to diagnose. When I say diagnose, I mean it's difficult to characterize what type of tumor it is. And secondly, if you are able to do that, say for example, then it might so happen that it might so happen, no, it is always very difficult to target that type of disease, because the more heterogeneous it is, the more difficult it is to figure out one way of killing those cells, right? It will have a lot of other subpopulations that will survive a lot better, so that becomes a much more difficult challenge to tackle. Okay, we'll move ahead, but before that, if there are any queries, please let me know. If it was clear, please let me know that as well, and then I'll move on.
No. Ma'am, no doubt.
Okay, cool. Moving ahead, we are yeah. We'll understand the definition of the two a little more clearly. So genetic mutations that drive the development of cancer are defined as driver mutations. All other mutations which play just a secondary role in cancer development are passenger mutations. I think I must have explained this previously also at some point of time, but I don't think I had a slide on it. But basically, yeah, these are the key differences. Also, keep in mind that driver mutations typically they are somatic mutations, which means that they arise de novo in cancer cell. If a particular mutation is hereditary, usually that is not considered to be a driver mutation. It is a it is probably going to be passenger. It might be a secondary driver mutation, which means that something else in that particular person has changed or has altered, as a result of which lacking a mutation or lacking the proper functionality of this particular gene is making the disease much worse for them. So therefore, typically driver mutations are considered to be somatic in nature. Now, this is showing you the emergence and selection of somatic mutations, how exactly they're eventually developing. So the first set or the first panel is showing you DNA lesions that are yeah, DNA lesions that are generated over time, and that is most commonly due to multiple cycles or very unchecked proliferation or very rapid cell division, as a result of which, during the process of DNA duplication and the process of segregation between the two cells, a lot of things go wrong. The polymerase efficiency goes down, as a result of which its ability to properly place one particular base as a complementary residue to the existing base, all of that is severely compromised. And as a result, what you see are different types of lesions that can be formed. Example being A, you can have DNA damage, you can have unrepaired DNA lesions that have persisted through several rounds of replication. You can have multi-allelic variation. Under multi-allelic variation, you can see that the overall, like the different microsatellite markers or different portions. Let's look at them as bases only. The order of bases between these three fragments is quite similar, However, they only lack difference in one or two bases or one or two regions, and therefore this variation is considered to be a multi-allelic variance because they are eventually all alleles of a particular gene. They lack very subtle changes or subtle differences between each other. So this sort of retention and eventually the propagation, which means the passing on of such genetic mutations, is something that leads to multi-allelic variation, and this is actually quite common as well. What you see here is that this damaged DNA can actually be used as a tracker. How so? Uh, Okay, let's look at this particular panel, which is panel B. So, initiating genetic mutations observed in cancer cells can be found in normal cells at the tumor site, allowing us to actually trace the origin of the tumor. So, at the tumor site, you can find these initiating genetic mutations, and so you can figure out where the tumor has eventually originated from. Third, in section C, you can see that the genetic instability during tumor progression leads to the acquisition and selection of some tumor advantageous variants. Variant selection can actually be studied by modeling the recurrence of somatic mutations across large tumor cohorts. Developing machine learning approaches to predict variant oncogenicity and investigating whether the selection of specific variants depends on either pre-existing alterations or on the cell origin of the tumor. Uh, what is the story overall? So first, you have accumulation of genetic damage or DNA damage, basically. Then that can be incurred or that can actually be present in cancer cells. And eventually, during this process of tumor progression, if a lot of genetic instability is present, then that can lead to this sort of selection, like we just discussed. And different sort of approaches are being put in place to predict variant oncogenicity to select whether they can actually give rise to a tumor and they can be actually considered to be um, a driver or a passenger mutation. So here you see you have a tumor advantageous, you have a population that has a particular advantage, and you can actually model this by modeling the mutation recurrence within a particular population. You can create that mutagenesis in silico, or you can look at pre-existing mutations and lineage constraints and then see how the tumor is gradually evolving. So all of these attributes are quite commonly studied and they're pretty well known. Then comes the idea of cell state. So, what is this cell state exactly entailing? Just okay. So, the existence of cancer cell states has many implications. First, the genetically identical cells might actually be phenotypically very heterogeneous, and therefore they can implement different sort of functions to foster disease progression and treatment resistance. Second, the treatment and microenvironment associated conditions are likely to sort of impact cancer cell properties and thus they drive state transitions. Third, opportunities they exist to reprogram cells to a therapy sensitive state and to test all of these different hypotheses. There's a need to develop models that recapitulate not only the genetic diversity in the tumor, but also the presence and transition of dynamic states. So, what does this basically mean? Okay. Okay. Yeah. So basically, what you can see are different sort of modifications that are undergone by these cells with a lot of toxicity. What does that entail? That entails epigenetic changes, like for example, modifications within the histone, Second, some modifications within the microRNA, interactions with the tumor microenvironment. They all can induce epigenetic and transcriptional reprogramming that eventually can alter the cancer cell identity. This process defines cancer cell plasticity conditions, transitions among phenotypically very heterogeneous cell states that can be inferred from single cell molecular profiles. So what does it meaning? It means that you are defining cancer cell plasticity as the transition from one state to another, which are quite heterogeneous. And this can actually most commonly be inferred from single cell molecular profiles. Examples of such cell state transitions have been reported in a couple of different types of malignancies. The first example given here is for skin melanoma or skin cancer or melanoma, where mesenchymal and stem-like states have been associated with metastatic and tumorigenic capacity, respectively. In lung adenocarcinoma, where disease progression was associated with morphological differences and transition from an alveolar type 2 like state to a dedifferentiated and proliferative cell state. And in breast cancer, resistance to therapy was shown associated with cell dormancy and random awakening mediated by epigenetic reprogramming. So that's what you basically see, that using single cell omics, which usually involves single cell RNA sequencing, most commonly that is what is involved, they have seen that there's a sort of difference or at least an overlapping transition from physiologically normal cells to this to the plastic cancer cells or cancer cells which have undergone a lot of modifications. And following that, you can see that different types of cancers have actually reported examples of the same. First example being melanoma. Second example for lung cancer, this has been observed, and for breast cancer as well. So yeah, that is pretty much on that slide. Any queries or any doubts, you can let me know at any point of time. Um, then we'll move on here. Just a second, I'll get some water. Okay, yeah. So then moving on, we are going to learn a little bit about the commonly established models for cancer evolution. So majorly there are two types of cancer evolution models that are known. What are the two types? The first is the clonal evolution model. The second is the cancer stem cell model. So for clonal evolution, what is it known as? It is known as a non-hierarchical model where the mutations that arise in tumor cells, they confer a selective growth advantage. So how do you actually understand that? So here typically you can see that from the first cell, first of all, you have to understand the fact that within each tumor, some types of cells are considered to be as stem cells and they're called as cancer stem cells or CSCs. Here, what you see is the first cell that has faced some sort of a change. Like you can see in red, this indicates some sort of a modification that has undergone. So that eventually has given rise to two types of progenitors, which has again faced some sort of, some sort of a mutagen exposure or some sort of changes. And then this in the next generation, you can see that this particular cell has completely changed from what it was supposed to, from what it was supposed to be compared to its peers. Let's look at it that way. So during the process of cell division, this particular cell has lost its identity and has produced a clone which is quite different from that of the others. So this particular cell, it would be considered to be a dominant clone. Why do we call it a dominant clone? That is because it has from its from its progeny, which means that when this particular cell divides and it generates two types of cells, one of them will actually retain the ability to keep on proliferating, which is indicated by this little sort of arrow, which is indicating the regenerative capacity. So that can give rise to a mature cell. Or what can also happen is that it can eventually lead to the formation of a large tumor. So what is being said here? Let's go back to the legend. So this particular cell shown here in red has acquired a series of mutations and has produced a dominant clone. Tumor cells shown in red and orange arising from this clone, they have similar tumorigenic capacity. Other derivatives shown here in gray, they may lack the tumorigenicity due to stochastic events. Tumor heterogeneity results from the diversity of cells present within the tumor. Okay, so that is the key difference between the clonal evolution model, which means that from the initial cell which was present, one particular clone has undergone a lot of mutations, and that actually generates more subclones. Basically, the diversity which is generated from the initial cell, among that population of subclones, some of them would be dominant, some of them would be less dominant, but technically they can all eventually lead to clonal evolution. They can all eventually lead to tumorigenesis, which means they can form a tumor. However, because there are different types of cells or different types of subclones which are present. Um, Keep in mind that here, just for the sake of understanding, only two types have been shown. But practically, when you think about a tumor and you think about the type of alterations that can be present in a tumor, we have talked about it before as well, that the amount of genetic heterogeneity which actually exists is huge. And as a result, that heterogeneity is supported by the fact that different subclones are present within the same tumor. So, for example, within this tumor, the red population might have mutation X, light red population might have Y, and 10 different types of mutations might actually be present. How is that advantageous? It's advantageous because when this particular tumor faces treatment with some sort of a drug, one of these populations is possibly going to have the ability to survive under that condition and survive better under that condition and outcompete all of the other different types of cells. And as a result, it is going to generate a tumor which is now treatment resistant. So from the all of the different types of subclones which are present, the populations which do not have this tumorigenic capacity, they are not going to survive. However, from these populations, those which are able to survive better and adapt better to the surrounding environment, they are going to take over and they are going to eventually form tumors. So that is part of the clonal evolution model, which means that it results from the diversity of cells which are present within the entire tumor mass. The second model, which is the cancer stem cell model, is predicated on a hierarchical organization of cells. So this is another key difference between the two. So for the clonal evolution model, it is a non-hierarchical model. This is a hierarchical model, and it depends on this organization of cells. So under this, a small subset of cells has the ability to sustain tumorigenesis and generate heterogeneity through differentiation. What does it mean? It means that they are undergoing modifications. They have the ability to be tumorigenic and they have the ability to generate or create more heterogeneity. And that is because they are stem cells. So among the population of tumors, the stem cell will have the ability to divide and will have the ability to generate more and more diversity. And in the example here, a mutation in the progenitor cell, which is depicted here in brown, which is this, has endowed the tumor cell with some stem cell-like properties. So these stem cells have self-renewing capability and they can give rise to a range of different tumor cells, depicted here as gray and green, thereby accounting for tumor heterogeneity. So these, when they divide, they can generate a lot more diversity, gray, green, and red. And among these, by itself, green might not be able to generate a tumor by itself, gray might not be However, when red is present, it can divide, which means that it is a cancer stem cell. Red can keep on dividing, and red can generate more and more types of gray and green, and it can keep on increasing the diversity even further. So basically, that gives rise to more tumor heterogeneity. Now, both of these models, while they might seem really similar, and they are overall appearance gives you quite a lot of similarity, but the fact lies that the difference between the two is coming from the fact that there is a stem cell which is involved. So if you have learned a little bit about stem cells, you would know that they have a couple of characteristic features. The first of them being the ability to keep on dividing, the ability to self-renew, which means that they can keep on producing more and more of themselves. Thirdly, to generate more the ability to generate all of the subtypes of that particular tissue. Like, for example, if you think about the hematopoietic stem cell, for example, it is a multipotent stem cell, which means it can generate all these types of blood cells. Every type of the blood cell can be derived from the progenitor hematopoietic stem cell. So like that, all of these properties are present, and hence the cancer stem cell model usually is more acceptable. Usually, of course, there are exceptions to that. And there are different models, different research articles which support the clonal evolution model as well. There is actually some studies which have correlated the two with each other. Clonal evolution and cancer stem cell model have actually been studied together, because see, in the long run, if you think about it, if you as a researcher think about it, you can you will have evidence that supports the fact that within the tumor there are certain cells which are stem cells. And secondly, you can also see evidence that from the population some subsets are actually getting selected better. um, but yeah, the very hard and fast definition of the two, or the a very hard and fast conclusion that yes, X model is right or Y model is right, is not till date something that is being debated. And it is believed, the overall idea that is followed and that is widely acceptable across the world is that A, cancer cells evolve, B, they adapt to the condition, C, they in a lot of conditions are able to actually support the clonal evolution model, whereby you can see that certain subpopulations which were already part of the parent population, they have survived and they have taken over. Oops. And secondly, the second hypothesis has also been observed about cancer stem cells. You'll find lots and lots of reviews about that as well. You will also find studies which give you n number of other types of models as well. These, the two that I'm discussing with you today, they are the most traditionally, like this is something you will find in textbooks. They're something that you will you find like in 20-year-old, 25-year-old papers also, you'll find these topics being discussed. However, recent studies have given you a lot more information, and a lot more subtypes of cancer evolution models have been proposed, which are intermediate, which might be completely different. So yeah, it's more complicated. It's not required in the scope of these studies, so we have avoided that. We'll look at these traditional two models. But yeah, basically the key difference between the two is the fact that there is a hierarchy in the cancer stem model. The uppermost creamy layer is supported by the cancer stem cell, and this model believes that within a tumor there are certain stem cells which are present, which are actually capable of generating more and more heterogeneity, and that is helping it survive and adapt better to the type of condition that is there. Whereas the clonal evolution model goes completely in the opposite direction and says that within a tumor there is a subpopulation that already exists, and no additional diversity is eventually arising. But in fact, from the present population, only one of them is getting selectively or is able to selectively progress and therefore overcome the challenge faced by the tumor as a whole. So yeah, that is the idea. Now, this actually leads us to the concept of drug resistance. So just a second. Yeah. So this is actually a quick snapshot of what like if you are looking at the world around you, if you are part of a tumor, if you are just a cell as part of a tumor, what are the types of challenges and what are the types of adaptations that you can actually undergo? This schematic helps you understand that really well. I think I forgot to put the reference for it. Um, yeah, maybe I'll add it later on. This is also taken from a review. Just can't remember which one. I'll add the reference. I have it. So you can go through any review to understand drug resistance. It's obviously a very easy concept to understand now that you've learned so much about cancer evolution. But basically, the idea is that in a timeline, if at point A, a particular patient is diagnosed with cancer, at time point B, if that patient is treated with some sort of a drug, in time C, usually remission is observed. However, at some time point D, there is a very, very high chance that eventually relapse might also be seen. That means that while the patient responded very well initially to the treatment that you gave, there is a very high chance that in the long run, that remission, like um, the remission, how do I define it? Remission basically means that the patient gets better. Uh, yeah, the patient gets better, but however, after a point of time, it might so happen that the patient again is diagnosed with cancer, and this time, obviously, the disease is a lot worse. And usually, these sort of patients, they usually do not make it because if you remember, I had discussed this previously as well at some point of time, that it happens very commonly, especially when the patient has undergone some sort of surgical treatment, because while the tumor might have been removed from the primary site of localized cancer, like for example, say for example, if a person suffered from uh, liver cancer, and the initial diseased liver was removed out, and that local area, was irradiated, etc. All of that was done. They might initially show remission, they might start responding very well to the treatment, and it might look like, cancer has gone away. But in more often than not, what is observed is that 1 year or so later, the patient starts seeing the same symptoms, and this time the disease becomes a lot worse because, at the time of surgery, it's possible that complete removal might not have happened, because obviously you think about it in this way. At the end of the day, the person performing the surgery is human. That's a trained expert human, but is human at the end of the day. And what you're removing is a cell, right? So some cell which has undergone some mutation so that it is also cancerous might not necessarily look like it is part of the tumor. As a result, the resection part, or the part of the organ which has been resected, that might look like you have removed the complete tumor, but it might so happen that some part of it has remained behind. Think of it like removing mold from bread, like so if you see that there's fungus on a bread and you remove the contaminated part of it, or any for that matter, it looks like you've removed the contaminated part of it, but the spores might actually be present throughout the fruit or throughout the bread. So if you leave it out again for some time, you might again eventually see the growth of the fungus. Pretty similar to that, something like that, and much worse happens in the case of cancer. So what looks initially safe might actually eventually develop into cancer, full-fledged cancer as well. So to actually bypass that, in a lot of cases, complete removal of organs is done, if it is, of course, not life-threatening. You cannot deprive a person of liver. That's not possible. If it is, say, for example, an ovarian cancer, then complete removal of ovaries is performed, oophorectomy is performed. Or if somebody has breast cancer, for example, that can be removed. You cannot remove, in a lot of cases, you cannot completely resect a part of somebody's brain. If somebody has a particular tumor in one part of their brain, you cannot remove the entire lobe, right? So it's these are practical challenges. So to tackle that, people usually go for other ways of treatment. So some sort of radiation, other therapeutic strategy, all of this is usually adapted, but of course they are not foolproof, and some metastasized cells might actually lie in other parts of the body, and they might not exactly show up as part of these scans that you perform regularly for a particular patient. So, say for example, even if I say regular, a patient cannot go for checkups every week, right? That's not something people usually go for. It's usually once in 2 months or 3 months. That's the quarterly treatment or quarterly follow-up that people usually opt for. So it might be missed, one of these tumors might be missed in one of the sessions, and then when the disease develops eventually in 6 months, it's going to be a lot worse, and therefore relapse is something You see really commonly, especially because during this entire time, that patient has also possibly also been on some sort of chemotherapy as well. So as a result, whatever has survived now is a resistant variant of the initial cancer, and therefore it's a lot more severe. And now you need to figure out some other way of treating that tumor to get rid of it. So yeah, things are a lot worse, and yeah, it's something you see very regularly in clinics, and it remains an open challenge till date. So these are this schematic actually highlights the different biological determinants of drug resistance. So, like we've discussed, tumors are heterogeneous. They're situated in a milieu that comprises the basement membrane, the vasculature, immune cells, and tumor microenvironment, among other components. So first of all, if you look at this mass, you'll be able to see different colors and different types of cells which are present, indicating the tumor heterogeneity. Then the fact that it lies on this basement membrane and it is surrounded by the vasculature, which is the blood vessel. It is surrounded by some sort of immune cells as well, like T lymphocytes, B lymphocytes, for example, and other components. All of this can actually be present. Changes in the physical parameters, the genome, and the surrounding microenvironment of the tumor actually can drive drug resistance. Like, for example, you can have some physical barriers to that treatment. The amount of tumor might be so huge that your drug is not able to tackle it. The tumor might be proliferating way faster than your drug can act, or the drug has been tolerated by the tumor, and as a result, the therapeutic pressure has led to a modified tumor, which is much more severe and much more resistant to the surrounding condition. Or it might have reprogrammed the surrounding immune system, which now supports its growth. Like you can see, it is surrounding the tumor and helping its growth instead of compromising the same. Or it might be some sort of a mutation or some sort of a change which is not druggable, and therefore the drug is not able to tackle this problem at all. So these are some of the common biological determinants of drug resistance. And what you see on the right is showing you the standard of care approaches, which means the most commonly followed approaches, and some emerging approaches to manage the biological determinants of resistance. They can be targeted by a number of clinical, diagnostic, and therapeutic strategies. So what can those strategies actually be? So on the right, you can see, first of all, tumor growth. How can you tackle that? This particular attribute. You have to subject that patient to dynamic monitoring. You have to perform functional imaging studies. If you remember all of those different imaging diagnostic tests that we talk about, like PET scans, we talked about bone scans and MRIs, fMRIs. All of these are, again, let me just reiterate the fact that these tests are very expensive, and let's talk about the practicality of the situation as well. So not everybody has the financial backup to support this sort of treatment, and B, uh, if you think about it in the long run, it is not feasible also. How do I explain it? If you ever have gone, unfortunately, I hope you haven't gone, but if you, as part of some, experiment or whatever, or internship, etc., if you eventually go to some cancer hospital, you'll see the burden that exists there, the number of patients that are suffering from the disease, and how practical it is to advise every sort of patient to go for very regular functional imaging studies. It's not possible. We, as a population, are so immense, and the subpopulation that suffers from cancer is so immense that priority wise, patients actually have to be categorized, and only then somebody can be subjected to some treatment. So you have to be financially well backed up. B, you're supposed to have access to all of these tests. And C, you have to be educated about it. Not everybody knows. How many people do you think know about cancer, and how you should actually progress with your prognosis? Not everybody knows that. People follow somebody's advice, and very directly speaking, the sort of medical infrastructure we have does not always support such a huge population. So all of this that I'm showing to you, that is all part of research and part of a population. Usually these are population from Western countries or from the European cohort. So there the population is so limited and so controlled, and the medical facilities are a lot more accessible, I wouldn't say affordable, but accessible in a larger scope of things, that all of this might be a lot more possible. Read about clinical trials. I think I told you this recently. Read a little bit about clinical trials. Do a comparison between a clinical trial from the West, say any of the FDA approved USA based trials for any cancer, and look for one in India. You'll see the stark difference between the two. Why so? Because enrolling a patient for a clinical trial and ensuring that they come back every 3 months or 6 months for follow up, and then ensuring that you can actually track how drug resistance is emerging and how a particular tumor responds to a certain treatment. Completion of that study by itself is going to be next to impossible in a country like India. However, the same is a lot more feasible in these other sort of countries. A, because here people don't turn up. B, if they start feeling better about the symptoms, they don't bother continuing with, the follow-up treatments. C, they don't have the financial backup for it. So they just opt for palliative care. They think that, if I have X amount of money, I have to invest it in something else. So all of these studies, they're a lot more compromised. And honestly, there's no direct solution for it. But being an active citizen of the country and being somebody who is learning about cancer, you should have the knowledge and you should have the ethical backup to figure out that if you know somebody who is subject to this sort of treatment, you can at least have the responsibility of informing them about all of this. And the rest is, of course, not in your control. However, let's come back to the ideal situation and learn about the different ways in which drug resistance and treatment can be managed. So yeah, these are the ways in which tumor growth can be monitored. Apart from that, tumor burden can be monitored by neoadjuvant chemotherapy, surgery, radiotherapy. Tumor heterogeneity can be tackled by early detection, by eventual combination therapy targeting tumor neoantigens. Physical barriers can be overcome by different methods, again, fMRI, etc. Just read about it. I think it's pretty much something we've already discussed. For undruggable genome, if you remember, we discussed this in the last class, where we talked about how transcription factors, which were previously considered to be undruggable, have now opened up their scopes in the recent past, and you can use inhibitors against those. Allele-specific inhibitors, restoring tumor suppressor function. This is also something that it seems very promising, but very difficult to implement. But yeah, it has a lot of potential. Therapeutic pressure, you have to figure out ways to overcome resistant mutations, allosteric inhibitors, antibody drug conjugates. Again, all of these are topics we have discussed at some point of time a few weeks ago, a few classes ago. So you can quickly refer to those for your own understanding. But yeah, these are the basic on-paper strategies which are used to tackle drug resistance in cancer. And this actually shows you another sort of timeline-based understanding of how you can figure out how cancer drug resistance is emerging and how well your patient is responding to the treatment that you're subjecting them to. So these are the three basic strategies. Yeah, this is the paper. I forgot to put it in the previous slide. I think it's part of the same paper, A View on Drug Resistance in Cancer, published in Nature in 2019. So the previous figure is also from there. So the first one is to figure out ways to detect cancer a lot early and to intercept it as well. So this strategy, which again is something we have discussed. So as you can see, the sequence of topics to be covered as part of this course was such that you become familiar with a lot of terminology by the end of it. So we have discussed all of these things so that ctDNA, like for example, as part of the non-invasive diagnostic techniques that we talked about, that is going to make it a lot more feasible to diagnose cancer and to intercept it as early on as possible. So ctDNA to detect cancers with or without effective screening, ctDNA to detect cancers that are virally driven, to detect therapeutic vulnerabilities, very, very important part of it, because if there is some sort of modification that the cancer population is undergoing, which is making it treatment resistant, then if you figure out a therapeutic vulnerability based on profiling of the circulating tumor DNA, that is probably going to give you a new angle to chase and a new way to tackle those treatment resistant variants, and a way to figure out some way of performing combinatorial therapy or just tackling the treatment resistant population. So yeah, so this is actually showing you that over a period of time, if you look at the tumor mass, so this is how the tumor was expected to keep on increasing, the gray line, what you see here at the top. However, if early detection can be performed, and at this point of time, if treatment can be actually started, you can see that decline can occur. Secondly, if you look at deeper response, this is, see, these are different ways in which you are trying to tackle drug resistance. It's pretty much going to be similar, but here you are using multiple monitoring stages and multiple ways of tackling the same. How so? First of all, you can see you are improving the therapeutic index. You are performing localized administration of therapies. Like, for example, if something which is, I'll give you an example of something which is done for prostate cancer. So there's something called as brachytherapy. So what is done under that is that little chips, like radioactive chips, small radioactive chips are placed directly in the area very close to the prostate gland, so that there is very localized, very targeted radioactive emission which is exposed, which the cells of the prostate tumor are exposed to. And you, by that, what you're doing, you are minimizing overall body spread of radioactivity. You are minimizing the number of cells which are exposed to that radiation. Its half-life is quite small. You're not unnecessarily subjecting them to a lot of radiation for an extended period of time, and you're also ensuring that there is very like the minimized spread of radioactivity can actually be ensured by that. So this is just an example of the same, using antibody drug conjugate so that you can deliver the drug a lot more in a lot more targeted fashion, and using more potent inhibitors directly instead of, say, for example, we talked about Taxols very early on, right? And we discussed how they are never the first line of therapy for any particular patient because it has a lot more off target effects. And it is something which is usually administered for a patient who has progressed to a much more advanced, much more untreatable stage, because the non-specificity and the side effects are a lot more events. So in this case, if early detection is diagnosing that a subpopulation of treatment resistant variety is emerging, then such a patient might actually be recommended to go for this kind of exposure to more potent inhibitors. So like you can see, the decline here is different. So multiple administrations are actually being performed. And so the using that early diagnosis, it is going to lead to a much quicker decline in cancer. It is going to make things a lot more manageable in the long run. And thirdly, therapeutic monitoring and adaptive interventions can also be put in place. So therapeutic monitoring basically means that after administrating the treatment, that patient will be monitored for the type of the response of the patient to that type of treatment would be monitored using non-invasive methods once again, which are ctDNA. So again, following the same graph, first an initial therapy can be administered. So here the diagnostic part is not being covered under non-invasive technique, but instead the response is being used to the response is being studied using those methods. So first, initial therapy is administered, and after that, adaptive treatment might also be eventually given to see what type of response is coming about in that patient. And all of that monitoring is being done using ctDNA. Furthermore, ctDNA is used to monitor and detect clonal evolution and see whether there is the emergence of resistant varieties. You try to figure out ways to generate combinatorial therapies. Of course, you figure out its response as well. Network modeling is done. It's beyond the scope of this. And also you can actually salvage the acquired resistance in real time because it's non-invasive in nature. So over time, every, say, a few weeks, somebody can be follow-up of that particular patient can actually be performed. And over time, how the tumor is evolving and how quickly it is evolving, all of that can be tackled, and accordingly, adaptive treatment can be performed so that their response to that treatment is also better, and there's a better chance of survival, leading to better prognosis, and overall patient response can be a lot better. So finally, to sum up, this was the idea I wanted to discuss for today. I kept it short, but yeah, anyway, we had less time, so that's worked in our favor. But overall, what we've discussed is, of course, that cancer is a very rapidly evolving disease. And remember, we talked about the fact that it follows Darwinian evolution, the idea of dynamic evolution, and it bypasses normal cellular control. Of course, this is something we have discussed extensively. By this, I would also like to bring your attention back to the hallmark feature, which is of genomic instability, a major contributor to the adaptability of these cancer cells, which leads to a lot more genetic heterogeneity. And of course, the other enabling characteristics are also important, which lead to lineage plasticity and also the interaction with the TME or the tumor microenvironment. Um, Furthermore, genomic instability also drives the rapid acquisition of drug resistance as well. However, under this, we have discussed two different types of models, both of which are hypotheses. Um, by models, you don't see, this is a difference between a model and a mechanism. A mechanism is something which has been experimentally validated, and it is, the only procedure or the only line of thought which you can expect for a particular pathway to follow. However, if something is questionable, which has multiple lines of thought, and there are experiments supporting and negating that, then that is considered to be a model. So these cancer evolution systems, they are still considered to be models. So take it with a pinch of salt. So that is what we talked about. And finally, several strategies, while they are being explored and drug resistance in oncology research is extensively being tackled. If you just perform like a PubMed search or even a Google search for that matter about cancer drug resistance and therapies and studies regarding therapies, it's huge. It's obviously a very urgent need, but also it's something that has persisted for a long time, still persists, and probably will persist in the future as well. So that will remain an area of extensive research in oncology. And if it's interesting, definitely a domain you should go ahead with. You will definitely have scope in it in the long run. Um, just some very basic takeaway questions. So, first of all, how has the field of omics opened up this arena of research for drug resistance in cancer? I did not highlight it, but read about it a little. If not, we'll take it up in our last class. And if necessary, again, if possible, I might take another extra class, or maybe we'll extend that. But I think 2 hours should be good enough for a discussion on all of the questions. But let's see. And finally, can we design or how can we design a foolproof strategy to bypass drug resistance in cancer? Think about it. Take it as a hypothetical situation. If that's what you were, if you were to design a strategy, what would you do? How do you think you can make it foolproof? Can you make it foolproof? And what is going to be the off-target effect or the potential of its application across different cancers? So just think of questions for you to ponder upon, and we'll of course have discussions about this. So with this, I'm done from my end for this class. Any queries, any doubts, please let me know, and then we'll wrap up for today. So please unmute and let me know. Yeah, Ankit.
Ma'am, could you please go to
Hello.
slide number 128? Hello. Yes, ma'am. Could you please go to slide number 128?
Sorry, there was some issue. I couldn't hear you. Can you repeat again?
Could you go to slide 128?
128. Yeah.
I mean, this cancer stem cell model, what
Mhm.
is the significance of that self-renewal thing?
That is a property of stem cells. So, like I discussed, there are some characteristic features. How will you distinguish a normal cell from a stem cell? So, one of its necessary capabilities is to self renew. So, for what does that mean? It means that if a particular stem cell is dividing into two, one of the progenitor cells, not progenitor, one of the progeny cells is going to be another stem cell, which means that that population will continuously be conserved and more daughter cells will be generated, which can have different potential. And in this case, in context of cancer, they are going to generate more and more heterogeneity, and they are going to make the tumor a lot worse. So it's a necessary capacity of a stem cell to be classified as a stem cell.
Okay, ma'am. And it is not in the progenitor cell.
No, no. So how do I explain? Take the example of hematopoietic stem cells again. So, and there's a very small population of HSCs or hematopoietic stem cells in your bone marrow. Okay. So what they have to continuously keep on doing is that they have to continuously replenish that population. It cannot become less, right? So they maintain a subpopulation of the hematopoietic stem cells. Some of it obviously has to keep on dividing so that you have RBCs, you have WBCs, you have platelets, etc. All of that is necessary to, it will keep on differentiating and it will generate the rest of the population required to sustain all of the cells within your blood. But some subpopulation has to be retained so that there is a HSC population within your cell. So actually that's what is done during like when a person suffers from leukemia, etc. When bone marrow transplant is performed, you basically restore or you provide the patient with hematopoietic stem cells present within their bone marrow because they've lost the ability to do so. So the HSCs become compromised. So that helps in which can now generate n number of cells, which will eventually divide and differentiate, and then they'll form all of those different cells. So it's a conserved population. Think of it in that way.
Okay, ma'am. Thank you.
Okay. Anybody else? Any questions? Okay, if not, then I'll end the class for today. So thank you so much for joining. We only have officially have two more sessions left. So we don't have a lot of content left to cover, so it should be fine. We only have the terminologies like cancer, statistics, etc. All of that to cover. That won't take a lot of time, I think. If possible, we'll start the discussion of the previous questions like from week one, so that we can split the recap between two classes. I think that will make it more feasible. So we'll do that, and then we'll wrap up the course. So any doubts you have had during the course, please record them and we can have discussions on them. Anything you did not understand at that point of time, or you did not attend the class, you only went through the slides or something like that, please bring that up. We'll try to cover it up. And yeah, we'll have the next class. I'll send an email, but we'll have the next class on Friday or 28th from 5 to 7 p.m. And the last class would be on 31st on Monday, same time. So that is the work plan. So please join in for the next class and we'll have discussions then. So thank you so much for joining. Have a good night. I'll end the session now. Thank you.
Thank you, ma'am.